Hello, everyone. Welcome to the second talk in our worldwide neuroscience, neurobiology of addiction series. Our speaker today is Dr. Meaghan Creed, and we're very excited to have her here. And to introduce her, we have Kurt Frazier, who will also be our co-host today. Go for it, Kurt.
All right, it's my great pleasure to introduce Dr. Meaghan Creed as this week's Worldwide in Neuroscience of Addiction seminar speaker. Dr. Creed completed her bachelor's with honors at the University of Toronto in Canada, and then she also stayed at the University of Toronto to complete her PhD work with Dr. Jose Nobrega, where she studied how DBS could be implemented in the basal ganglia to affect rodent models of movement disorders. She then journeyed across the Atlantic to do her postdoc in the lab of Dr. Christian Lucier, and while there she studied a wide variety of topics, including plasticity induced by cocaine exposure within the mesostriatal system, as well as identifying a potential novel treatment to apply DBS in combination with pharmacology to reverse drug-induced synaptic plasticity within the nucleus accumbens. In her own lab at the University of Washington, at the Washington University of St. Louis, Dr. Creed is interested in how pain and drug exposure cause synaptic plasticity within reward circuitry, with an acute interest in how, an emerging interest at least, in how the ventral pallidum is affected by these processes. Today, Dr. Creed is going to talk to us about two of those projects in her lab, one focusing on a novel subset of ventral pallidal neurons, glutamatergic neurons, as well as a novel projection that their lab has recently identified in controlling reward consumption. So if you have questions during the talk, please use the Ask a Question feature, and then we'll poll those, and you can actually vote on them, and we can choose those questions for Dr. Creed at the end of the talk. And with that, Dr. Creed, you can take it away.
Great. So thank you so much to Marina for hosting and for Kurt for the kind introduction. I will say that this is the second talk in the series of the neurobiology of addiction, and in some ways that's great. So last week we heard from Patricia Janik, who gave us a really elegant overview of the ventral pallidum, and you're going to be hearing more about the ventral pallidum, or the VP, today. And in other ways it's not so great because she set the bar incredibly high, and I've got very big shoes to fill. So thank you for attending, and as Kurt said, I'm going to tell you a little bit about our lab's recent focus on the ventral pallidum and its reward-seeking behavior. And I like to start my talks with this slide, which really gives an 1,000-foot overview about why we care about reward circuits. And what this is showing you, this is my very high-level synthesis of a dozen or so meta-analyses, just underscoring the comorbidity between chronic pain, mood, and substance use disorders. So what you're seeing on this axis is the primary condition that a patient would present with, chronic pain disorders, mood disorders, or substance use disorders. And then on this axis, you're looking at the proportion of patients that also meet criteria for a comorbid mood or substance use disorder. And what you can immediately appreciate is that of patients who present with chronic pain, almost half of those patients will also meet criteria for a mood disorder, and over a third will meet criteria for a substance use disorder. Strikingly, at least when I started digging into this, is that the association also goes the other way, with patients presenting with mood disorders. Over half of these patients also meeting criteria for some sort of chronic pain disorder, with a really incredible odds ratio. So your risk of being diagnosed with a chronic pain disorder being 7.1 times higher if you have a mood disorder as well. So the main point that I hope you can appreciate is that the extent of comorbidity between these conditions is really huge. And we're interested in studying, and while these comorbidity can arise from common genetic or environmental risk factors, we're really interested in the fact that these circuits are also, these disorders involve overlapping neural circuits, or dysfunction of overlapping neural circuits. And so really the reason we're studying these neural circuits is to try to understand how these different disorders come into play, how they induce plasticity in the reward system, and how this adverse plasticity in the reward system leads to maladaptive behaviors that are common across the spectrum of disorders. And so as Kurt mentioned, the circuit that I'm going to talk to you about today is really the ventral basal ganglia. This is a highly simplified rat's nest kind of view of that structure, where you have dopamine neurons within the ventral tegmental area projecting up to the medial prefrontal cortex and the nucleus accumbens. And then according to classical models of the ventral basal ganglia, you have the nucleus accumbens, which integrates excitatory executive information from the medial prefrontal cortex. It's not shown here, but also contextual information from the hippocampus, and limbic or valence-related information from amygdala-like structures, and it integrates this information and passes it downstream to the ventral pallidum, which then, according to classical models, serves as this limbic motor interface, translating this information or input from the accumbens into motor output through its projections down to the thalamus and brainstem. And my personal interest in the ventral pallidum really started when I was a postdoc in Christian Lucher's lab, where there'd been an extensive work looking at drug-induced adaptations within the accumbens, but relatively little work looking at what happens to plasticity at the output of those accumbal neurons. And I'm not going to show you any hard data from this paper since it's already been published for 5 years now, but I do want to just summarize what we found. That was, if you're looking at the output of the accumbens to the ventral pallidum, one big difference between the dorsal basal ganglia, where you have this clear segregation between the direct and the indirect pathway, is that within the ventral basal ganglia, we found that both the D1 direct pathway neurons from the accumbens and the D2, or the indirect pathway neurons from the accumbens, really impinged on a majority of ventral pallidal neurons, which was shown as well by Jonathan Kupchik working with Peter Kalibas, that this segregation of the direct and the indirect pathway really isn't valid for the accumbal to ventral pallidal projection. And in a nutshell, what we found is that exposure and withdrawal from cocaine treatment completely occludes activity-dependent plasticity at both of these projections. So we noticed that following cocaine exposure and withdrawal, that D1 inputs to the ventral pallidum were strengthened, and this occurred with a concomitant decrease in the synaptic efficacy of the D2 MSNs, that the D2 outputs to the ventral pallidum were weakened. There was extensive work at the time showing that potentiation of excitatory drive onto the D1 MSNs, onto their cell bodies, was related to cue-induced reinstatement or behavioral sensitization to cocaine. What we found by optogenetically reversing this plasticity was that modifying the D1 outputs to the accumbens was also related to the ventral pallidum, was also related to behavioral sensitization. And then I'm not showing you any hard data here, but Veronica Alvarez had shown that this altered drive from the D2 MSNs onto the ventral pallidal neurons was involved in the motivation for cocaine seeking acutely, whereas we found that following withdrawal, some amotivation for seeking of natural rewards was also linked to this altered plasticity at the D2 pathway to the ventral pallidum. And the reason we think this is because the D2 MSNs are an important source of endogenous opioids into the ventral pallidum, which are known to be antidepressive. So this has really sparked my interest in understanding the ventral pallidum and its role in drug adaptive plasticity, and really pointed to the ventral pallidum as a site of convergence for both the pro-sensitizing effects of addictive drugs and a potential interesting site for the symptoms of apathy or amotivation that emerge in drug withdrawal. And one of the things that really became clear when I was doing recordings for these experiments is that there's a huge amount of heterogeneity within the ventral pallidum, both in terms of the morphological properties of the different ventral pallidal neurons and in terms of their electrophysiological properties. And so what I really want to tell you about today in my talk is work that we've been doing in the lab trying to dissect some of this functional heterogeneity within the ventral pallidum, both in terms of the anatomical level and in terms of the neurochemical identity of these disparate ventral pallidal populations. And so specifically, as Kurt mentioned, I'm going to tell you a little bit about glutamatergic neurons that we've identified in the ventral pallidum and their role in adaptive behavior, and more recent work looking at archipalatal neurons, or these neurons that, instead of acting as a way station, as Patricia Janik put it, actually project back to the nucleus accumbens and play a really important role in orchestrating accumbal activity in the context of reward seeking. And so one of the main differences, or one of the many differences between the ventral pallidum and the dorsal globus pallidus, is that within the ventral pallidum, it's been reported for decades that there's expression of mRNA for vGlut2, which is the vesicular glutamate transporter. And so Jessica Tooley, when she was a technician in the lab, started to answer this really simple question. So we know that vGlut2 is expressed in the ventral pallidum. Are these vGlut2 neurons a distinct population from the classical inhibitory ventral pallidal neurons? Or are they potentially just a subpopulation of VP neurons with the potential for GABA and glutamate co-release? And she was assisted with this by Lauren Marconi, who was a technician at the time, and Jason Alipio, who was a rotation student. So the question was, are glutamatergic neurons a distinct subpopulation? And one of the ways that we answered this was simply by performing multiplexed in situ hybridization, looking at mRNA expression of VGlut2 and of VGAT, vesicular GABA transporter, which is a marker of GABAergic neurons. And when we looked in the ventral pallidum, we saw very little overlap between these two populations. It was less than 4% in our hands. And this has been replicated and found independently by Laurent Faget, working as a postdoc in Tom Nasco's lab. So now that we've established that these neurons are a distinct neurochemical population, we wanted to ask how these neurons modulate the mesolimbic circuitry. And to do this, we injected Cre-dependent channel rhodopsin into the ventral pallidum. And we did this in mice in VGlut Cre mice that express Cre only in those glutamatergic neurons. And we did patch clamp recordings in the lateral habenula, in the ventral tegmental area, and in the rostromedial tegmental nucleus, which were where we saw the densest projection targets, looking at the outputs of these cells. And if we did patch clamp recordings, we see that in all of these areas, we saw light-evoked excitatory currents in the postsynaptic cells. So this was true in the ventral tegmental area, and it was true whether we were patching onto putative dopamine neurons or putative GABAergic neurons. It was also true in both the habenula and the RMTG, and actually the rates of connectivity were slightly higher onto both of these structures. But what you might already see is that when we're looking at the circuitry, there's a lot of interconnected functional connectivity between these different brain regions. So habenula projecting to RMTG and to VTA. And when we're taking slices, particularly when we're taking coronal slices, we're severing all of this interesting connectivity, and we could be really losing insight into the circuit dynamics if we're performing our experiments only in slices. And so to answer this question, or ask how glutamatergic neurons affect plasticity in an intact network, we repeated the same experiment, this time injecting Phlox channel rhodopsin into the ventral pallidum. But instead of taking slices and recording from these areas, we lowered silicone probes into the lateral habenula, VTA, or RMTG. And we asked how activation of these glutamatergic VP cells affects firing activity within these nuclei when we have an intact functional network. And our results largely corroborated our slice physiology results. So when we were recording from the lateral habenula and we stimulated the glutamatergic cell bodies, we found that of all the neurons in the habenula that we recorded, over half of these were significantly modulated by light activation. And of all the neurons that were modulated, all of these neurons showed an increase in their firing rate in response to blue light. We saw a similar story with the RMTG, where again, about half of the neurons that we recorded from were significantly light modulated, and the overwhelming response was an activation, so that we've concluded that the VP activates both the habenula and the RMTG neurons. But our results with the ventral tegmental area were a bit more confusing, or a bit more nuanced. So what we found is that, first of all, there was a lower proportion of neurons that were modulated. And then within these modulated units, we had equal proportions that were decreased by blue light activation or increased their firing rate. And you can see that spread here. And we're not doing any optotagging or using any neurochemical identification here, but what was interesting is that of all the neurons that showed a decrease in the firing rate, they all exhibited low basal firing rates and had this triphasic waveform. Whereas the neurons that were activated in response to blue light, we never saw a triphasic waveform. We had narrower waveforms, and the initial baseline firing rates were higher. And what we're tentatively concluding here is that basically because of the network, the recurrent network connectivity, is that activation of the glutamatergic ventral pallidal neurons, even though there are monosynaptic excitatory projections to the ventral tegmental area dopamine neurons, is that due to these network effects of activation of the habenula and the RMTG, the net result is actually a functional inhibition of these dopamine neurons in vivo. And so what I've shown you so far is that these glutamatergic ventral pallidal neurons are distinct from the canonical GABAergic or inhibitory populations, and that they actually drive firing of the habenula and the RMTG and inhibit dopamine neuron firing. And this was important to us because the habenula and the RMTG together both are known to robustly encode aversive stimuli and to be important for punishment learning. And so we next asked what the contribution of these glutamatergic ventral pallidal neurons might be to reward-related behavior. And to do this, we started very simply with an optogenetic assay. We know from classical studies dating back to the 60s that mice will press levers for electrical stimulation of the ventral pallidum. So the ventral pallidum supports intracranial self-stimulation. And we just sought to sort of repeat that study, but using optogenetic tools. So we injected channelrhodopsin into the ventral pallidum and performed a real-time place preference test, allowing the mouse to explore two sides of this compartment. And we found when we stimulated the ventral pallidum with optogenetics, this is non-specifics or activating all of the ventral pallidal neurons, we repeated the classical electrophysiology or electrical stimulation results. And we found that mice prefer to spend time on the side of the chamber that was paired with optogenetic stimulation. So mice are preferring to have their whole VP stimulated versus spending time in a neutral chamber. And we didn't see this effect in our EYFP controls. Conversely, when we repeated the experiment, but now using our V-glut-CRE mice and only infecting the glutamatergic cells with channelrhodopsin, we saw the opposite effect, where now mice are actively avoiding spending time on the side of the chamber where they get their glutamatergic neurons stimulated, which is entirely consistent with this idea that the ventral pallidum, these glutamatergic neurons are exciting the RMTG and habenula, which are known, that excitation of these structures is known to be aversive. So we have this idea that stimulation of the glutamatergic neurons itself is aversive, which is in contrast to the gestalt role of the ventral pallidum, or the electrical stimulation. And given the role of the ventral pallidum in promoting appetitive behavior, we next hypothesized that these glutamatergic neurons might also play a role in modulating appetitive behavior. And to test this, we trained mice. Oh, I lost my mouse. We trained mice to lever press for sucrose under a really simple schedule to acquire this fixed ratio responding. After they'd acquired the task, we did baseline testing where we required mice to press a lever for 5 times in FR5 before each pellet of sucrose. And these trials were interspersed with progressive ratio sessions. And during the progressive ratio sessions, the requirement for the number of lever presses for each successive sucrose pellet increased. And that's what that looks like here, just looking at the FR5 lever presses. This is capped at 30 rewards. So we see a stable level of lever pressing at baseline in both the CRE positive and the negative mice that I'm going to show you. The session time, so the amount of time it takes them to reach their 30 rewards, is also stable. And then if we look at the breakpoints in a progressive ratio task, we do see discrimination between the active and the inactive lever, and again, a stable breakpoint. So at what point will the mice stop responding for sucrose? When the lever press requirement gets too difficult or demanding. So after we had this baseline testing, we then went in and took a sledgehammer approach and inject a CRE-dependent caspase virus bilaterally into the ventral pallidum. And this is showing you the ventral pallidum of a mouse that had just received a PBS injection. And you're seeing glutamatergic cell bodies co-expressing M-cherry within the ventral pallidum. And here you're looking at the ventral pallidum of a mouse that's received a caspase injection. So you have some remaining cell bodies and remnants of cell bodies and neurofibrils, but we're really inducing apoptosis robustly throughout this population and taking those cells offline, again, in a very sledgehammer approach. We gave the mice 4 days to recover and then asked them how they performed on these same assays. And we found no difference in the amount of lever presses in the FR5 task, so they're still just as efficient with their reward-seeking behavior. However, it took them significantly less time to reach their 30 rewards in their session, so the vigor of their responding and reward consumption is increasing after we take these neurons offline. But what was most striking was when we looked at the progressive ratio task, and we looked at the breakpoints, we saw nearly a doubling of the breakpoint, which reflects their motivation for sucrose responding after we take these neurons offline. And you can see that here in terms of the breakpoints or the active versus the inactive lever presses, where again we see a nearly complete doubling of the breakpoint. This is a really striking phenotype. The technician, Jess, who was running this at the time, she had started the mice at 8 in the morning, and she sent me a text at 10 p.m. one night and asked when she could stop because we had a single mouse that had pressed over 5,000 times for a single pellet of sucrose, which was really striking. So it's a really robust phenotype. And we wanted to know whether this was related to, the mice being just more motivated for reward, or is this more an insensitivity to the cost associated with reward? So we tried to get at that by taking mice that had already had caspase lesions of the glutamatergic cells and just letting them freely consume sucrose during a free access paradigm. And we see already at baseline that our caspase lesion mice consume more sucrose. But what was really striking here is that on the 4th day, we paired the sucrose administration with an IP injection of lithium chloride, which induces visceral malaise in these mice, and then asked how much sucrose they wanted to drink the next day. And as expected in our control mice, they all significantly attenuated their sucrose drinking following the lithium chloride pairing. And this was not at all true for the mice where we lesioned the V-glut neurons of the ventral pallidum. And in fact, many of them went back for more the next day. So they really weren't modulating their behavior in response to this punishing stimuli. And I'll say that this has been shown much more elegantly, more recently, by Marcus Stephenson-Jones working as a postdoc in Bo Lee's lab, where they trained mice on a head-fixed task where they were given increasing volumes of rewards or increasing reward values. This was water droplets in a head-fixed task where the mice were water restricted. See that the response reward goes up and scales monotonically with reward value. And in certain trials, they introduced an air puff punishment that was paired with reward consumption. And those are the conflict trials seen here. And you can see is that there's a downward shift in the level of responding at all reward values, and an air puff punishment was introduced to the reward. And then they went in and used the inhibitory opsin, archipalatal in glutamatergic neurons, the ventral pallidum, and stimulated these during these trials. And what they found is dashed line is the same data that you're seeing here, is that even when there was air puff punishment on board, these mice that had their V-glut neurons inhibited were behaving like mice like they had before there was any conflict, before there was any punishment introduced, suggesting that they're really not modulating their behavior in response to the punishment. And in fact, responding for reward actually goes up at the low values. Consistent with what we see, and really corroborates this idea that these pallidal neurons are important for adaptively constraining reward seeking, or for constraining reward seeking in the face of punishment. So what I told you in the first part of the talk is that these glutamatergic ventral pallidal neurons are a unique neurobiological population, that they activate the punishment network, and by doing so, they play an important role in adaptively constraining reward seeking. And now these glutamatergic neurons we consider non-canonical because of their neurochemical identity. In the next part of the talk, I want to tell you about more recent work where we've looked at neurons that are considered non-canonical because of their anatomical projection. So here we're talking about ventral pallidal neurons that project back to the nucleus accumbens. So as I said, these are considered non-canonical because classically the ventral pallidum is considered to be exclusively downstream of the accumbens. The model of information flow in the basal ganglia being that accumbal information is integrated from these executive and limbic structures, and that the ventral pallidum is more of a limbic motor interface, translating this input from the accumbens to downstream brainstem and thalamic areas. However, we were inspired by work from Nico Male when he was a postdoc, when he was a trainee with Peter McGill, where he described these archipalatal neurons in the dorsal globus pallidus. And so this is just a representative image from one of his beautiful papers where he's doing in vivo recordings, acute recordings of individual globus pallidus neurons, filling them with neurobiotin, and then reconstructing them post hoc to actually trace the projections. And this is an example of a prototypical globus pallidus neuron. So the cell body that was recorded from, and then tracing the collaterals projecting downstream to the entopeduncular nucleus, or the homologue of the internal globus pallidus, and onto the subthalamic nucleus and nigra. Then in the subset of neurons, he found these non-canonical external globus pallidus neurons, which he called the archipalatal neurons. And he named these neurons such because they project back to the striatum, but they do so in a way that sends these dense nets of collaterals over the striatal cell bodies, and these are two examples shown here. The archi being Greek for hunter's net, so they were named for their morphology and for their modulation of striatal activity. And as a postdoc in Josh Burke's lab, he went on to show that these neurons play an important role for sending stop signals to the striatum, so inhibiting striatal activity to cancel ongoing actions. And so we started to think about, if an analogous organization exists between the ventral pallidum and the nucleus accumbens, under what conditions is inhibiting the nucleus accumbens adaptive, or under what conditions do we see nucleus accumbens inhibition? And this is showing you a meta-analysis from Tanisha London when she was with Lex Kravitz, where she did a forest plotter. This is a forest plot from a meta-analysis, that she did, looking at several in vivo behavioral studies where they were recording from the nucleus accumbens while an animal consumed reward. So this is food and drug rewards mixed in here. But what you can see is that across all of these studies and across a variety of different paradigms, the proportion of accumbal neurons that were inhibited directly or specifically during the reward consumption is about 72%. So that the overwhelming or the majority response of accumbal neurons during reward consumption is an inhibition. As I mentioned, this was conducted across a variety of natural rewards and drug rewards and different behavioral paradigms. And so again, Jess Tooley, now that she's a PhD student in the lab, really sought to replicate this result, but using a totally unstructured reward-seeking task or a paradigm. So there's no organization to this, no structure to this task. There's no lever presses, there's no cues. It's really just free access reward consumption. And what she did was implant recording arrays into the nucleus accumbens and just freely let satiated mice explore this arena and freely consume a highly palatable rewarding solution. And she found that even in this unstructured task, if you align the neural activity of the accumbens to the start of lick bouts, she also saw this inhibition of accumbal firing. This is the average Z-score of all of the inhibited accumbal units, showing this inhibition. And to summarize this, just confirming that the inhibition of accumbal firing was the predominant response that she observed in her recordings. There were some excitations, which I'm happy to talk about in the question period and what I think that might mean. But she really repeated this idea that accumbal inhibition occurs with reward consumption, even in unstructured tasks. And then Ivan Bashes, who's a postdoc in the lab, took this one step further and tried to establish causality for this pause in promoting reward consumption. And there's already precedent in the literature that this pause in accumbal firing is necessary and sufficient. But he wanted to repeat this in our own hands, again in this unstructured task. And he did this by using the inhibitory opsin, archirhodopsin, and he injected it bilaterally into the nucleus accumbens. This is not a Cree-specific manipulation, so it's hitting all accumbal cells. And then he put mice in the same paradigm, just a free access task, except now when the mice were licking for their highly palatable reward, they had sessions where there was no stimulation, and they had sessions where this drove inhibition of their nucleus accumbens neurons by activation of green light. And he found no difference between the stimulated and the unstimulated sessions in the EYFP controls, but in the mice that expressed ARCH in the nucleus accumbens, that this closed loop inhibition dramatically increased the total drinking time relative to the unstimulated sessions. And this was related to an increase in the ongoing bout length and wasn't due to deriving more reward approaches, for example. So just confirming or showing these confirmatory results that inhibition of the nucleus accumbens is both necessary and permissive for reward consumption. And we were intrigued, thinking back to our ventripotent circuits, is that we know from a lot of elegant work, some of this has been shown, we heard about in last week's talk from Dr. Janik, is that the ventral, but it's also true in human patients as well, using bold fMRI studies, is that activity within the ventral pallidum tracks the ongoing hedonic value of reward, and it can do so on really fast timescales, so on a trial-to-trial basis, even when there's no learning or conditioning of the reward involved. And then what's really intriguing, this is from one of David Ottenheimer's papers with Jocelyn Richard, when they compared the latencies of value signals in the nucleus accumbens and the ventral pallidum, they found that value-related signals in the ventral pallidum actually proceed or occur with shorter latencies than those signals in the nucleus accumbens. And Ilya Monosov, who's a colleague at WashU, has also seen this and published this in non-human primates, that value signals arising in the ventral pallidum actually occur with shorter latencies relative to value signals occurring in the nucleus accumbens. So it really argues against the idea that the ventral pallidum is simply an output structure of the nucleus accumbens. And we hypothesized that this ventral pallidal, the ventral pallidum might be an important substrate of this accumbal inhibition that's both necessary and permissive for reward consumption. And to test this, we did some functional anatomy. And we did this by injecting channelrhodopsin into the ventral pallidum, but this time we took slices of the nucleus accumbens. And within those slices, we recorded from identified accumbal neurons. And we used transgenic mice that expressed fluorophore in different populations of accumbal neurons. So for example, here you're seeing a chat neuron that expresses TD tomato. So we can record from identified cholinergic cells. And here you're seeing fiber from the ventral pallidum that's labeled with channelrhodopsin that's in close opposition to the cell body. And we have transgenic lines that allow us to record selectively from parvalbumin interneurons, as well as D1 projection neurons and D2 receptor expressing projector neurons as well. So we have 4 populations of accumbal neurons that we can record from. And if we flash blue light over our slice, we can ask whether those ventral pallidal neurons are sending monosynaptic connections onto these identified postsynaptic targets in the accumbal shell. This is the underwhelming summary figure that we see here, where in fact we observe that over 80% of our D1 and our D2 neurons received monosynaptic innervation from the accumbal shell. Somewhat surprising to us, we saw that the interneurons, both the cholinergic interneurons and the parvalbumin interneurons, also received innervation, although this is a lower proportion. This is about 25 and 30% of cells, respectively, of interneurons that received innervation. And this is showing the location of each of these individual cells. So what you're seeing here is we patched over 100 cells in each of our genotypes. And what you're looking at is a compressed coronal version, or compressed coronal slices showing the anterior commissure, the accumbens core, the ventral midline, the midline, and the ventral surface. And we expected when we started this that we might see subterritories that were more functionally connected or less functionally connected. This is based possibly partially on Daniel Castro's work with Ken Barrage, showing that there are hot spots within the accumbens that are particularly important for generating this hedonic feeding. Or Reem Hassani's work with Michael Brukish, showing ventral versus dorsal subdivide in the function of D1 MSN specifically, with appetitive behavior being related to the more dorsal sections and aversive or avoidance behavior being more related to the ventral D1 expression. However, what you can appreciate from these maps is that we really didn't see any systematic differences in the connectivity between the subregions of the nucleus accumbens. So we found monosynaptic inhibition from the ventral pallidum onto D1 MSNs, onto D2 MSNs, onto parvalbumin interneurons, and onto cholinergic interneurons, really throughout the nucleus accumbens shell. And we know these currents are inhibitory because they were significantly attenuated, if not completely abolished, by picotoxin, which is a GABA-A receptor antagonist. And they were not affected by the combination of NBQX and AP5, which block the NMDA and AMPA receptors to inhibit excitatory transmission. So, like I told you about in the last part of the talk, we think that these glutamatergic cells are have prototypical projection targets, whereas these archipalatal neurons are exclusively GABAergic, and those two non-canonical populations really aren't overlapping. And we did a follow-up experiment, just again knowing that there's extensive reciprocal connectivity within the accumbens shell. And we wanted to ask if we stimulated the ventral archipalatal neurons or their terminals of the ventral pallidum in the accumbens shell, do we still see an inhibition or a functional inhibition of accumbal neurons in vivo? And to answer this, Jess Tooley implanted her electrodes over the nucleus accumbens shell, and injected channelrhodopsin into the ventral pallidum. And then she was stimulating the ventral pallidum fibers through an array that was co-implanted, through an optic fiber that was co-implanted with the electrophysiology array. And she recorded individual single units and multi-units, which she classified as putative medium spiny neurons based on their firing frequency and waveform, putative interneurons, and multi-units, where we couldn't segregate whether they were MSNs or interneurons. But what was really clear across all of these classes of accumbal neurons is that the overwhelming response to ventral pallidal activation or activation of ventral pallidal terminals in the accumbens was an inhibition of firing rate. So these are all single unit examples, but it was also clear at the population level, where the MSNs, the interneurons, and our multi-units all exhibit this robust increase in firing during ventral pallidal stimulation. And so if we know that these ventral archipalatal neurons are important for inhibiting the accumbal shell, we wanted to ask whether this ventral archipalatal activity was sufficient to drive reward consumption. And based on this hypothesis, we made 3 predictions. The first prediction would be that endogenous activity of these ventral archipalatal neurons would occur with reward consumption and might predict reward consumption. And so optotagging these neurons turned out, our efforts were really not successful, possibly due to our sparse population. So the way we tried to measure the activity, at least at the population level, was by doing calcium, measuring fluorescent signal with fiber photometry. So we did this by injecting a retrograde Cre virus into the nucleus accumbens shell, and then a Cre-dependent GCAMP, which is a genetically encoded fluorescence indicator, calcium indicator, into the ventral pallidum. And this is shown, and by implanting a optic fiber over the cell bodies in the ventral pallidum, we can selectively measure the calcium signal from these accumbal projecting VP neurons. And this is an example of a GCAMP trace shown here. And we did all of our experiments with equal numbers of matched GFP controls. And if we have our experiments, and if we do, is there a question? And if we have our mice performing this free access task and we align the GCAMP signal to the onset of a reward consumption or a lick bout, we see consistent with this early signal, we have an increased ramping of the calcium activity prior to reward consumption onset, and the peak of the signal occurs immediately after the reward consumption. So it's consistent with our hypothesis that the endogenous activity co-occurs with reward consumption. But then we asked whether this calcium signal can tell us anything about the behavior. So this is really tricky to decode with fiber photometry, especially on an individual trial basis. But the way we tried to answer this was for each mouse, we took the top quartile of trials where the GCAMP signal was highest, and the bottom quartile of trials where the GCAMP signal was lowest. And that's what this looks like here across all mice. And this is being calculated based on the initial 2 seconds of reward consumption, and we're taking the area under the curve. So this is just showing that we're splitting, for each individual mouse, splitting them into their high GCAMP and their low GCAMP trials. And now that we've split the trials based on the GCAMP signal, we can ask what the behavior looks like in those trials and whether this signal immediately after reward consumption predicts anything about behavior. And in fact, it does when we look at the probability of the mouse engaging in a lick bout. So here, this is the behavior now, not the signal. It's the licking behavior aligned to the onset of the lick bout. And what we see is that in these trials where we had the high GCAMP signal immediately after reward onset, if we look 10 seconds after the initiation of the lick bout, the odds are more than 50% of the trials, the mouse is still engaged in licking 10 seconds out. However, if we take the trials where they showed the lowest GCAMP signal immediately after reward consumption onset, almost immediately within seconds, the mice have aborted their trials in over 50% of cases. So we do see this different behavior depending on the initial area that was correlated with the initial area under the curve of this GCAMP signal. So this is one piece of evidence. We did the really simple controls to ask, especially based on this early ramping signal, whether this was maybe related to just reward seeking or reward approach. And we don't think it is, because if we introduced a requirement for an active nose poke before the mice could go get their reward, we didn't see any archipalatal calcium signal in response to the active poke or the actual seeking behavior, and we didn't see any calcium response in response to the inactive nose poke either. Again, telling us that this is really a signal that's related more specifically to the reward consumption rather than the seeking per se. The second prediction that we made was that if we activate these ventral archipalatal neurons, that we would be able to promote reward consumption. And we did this by, again, injecting our channelrhodopsin into the ventral pallidum and stimulating only the terminal fields in the nucleus accumbens. And after we had trained mice or habituated them to this task for several weeks, we started interspersing sessions where the mice either received no stimulation, where they received closed loop stimulation, where the stimulation was delivered at 20 hertz during the lick bout, or we had this open loop or scrambled stimulation where we delivered the same amount of stimulation as the closed loop trials, but we did it randomly throughout the session, completely independent of behavior. And so what we saw is that relative to the non-stimulated conditions, we didn't see any difference in the total drinking time with our open loop stimulation. However, when we did our closed loop stimulation, this is the EYFP controls and the channelrhodopsin mice, when we did our closed loop stimulation, we saw a robust increase in the total drinking time during the session. And this was due to a selective increase in the individual bout length, and it was not due to increased approaches or increased numbers of drinking bouts. So this was really striking. So especially thinking about the open loop stimulation, which had no effect on any of these parameters measured. So we didn't see any effect of open loop stimulation on the number of drinking bouts, which tells us that stimulating these neurons isn't sufficient to drive or to initiate reward consumption, which is consistent with recent work, for example, from John Britt, showing that excitatory drive into the accumbens, or the attenuation of that excitatory drive, is really important for driving the initiation or the reward approach. But that the inhibition or the information that the ventral pallidum is conveying is really promoting or prolonging that existing or that ongoing reward consumption. And just to get a little bit more granular with this, if we looked at the distribution of all of the individual lick bouts from all of the mice for all of their stimulated and closed loop trials, we can just see a little bit more clearly that the archipalatal stimulation is shifting the distribution away from these incredibly short bouts and promoting or shifting the distribution towards longer drinking bouts during these sessions, whereas we don't see any change in the distribution with our EYFP controls. And our final prediction, if the ventral pallidal activity is important for promoting reward consumption and accumbal-related inhibition, is that if we inhibit the ventral pallidal neurons, we should be able to attenuate the accumbal inhibition during reward seeking, during reward consumption, and that this should translate into attenuated reward consumption. And to do this, we did not want to use any terminal inhibition due to the biophysical constraints of existing optogenic approaches for terminal inhibition. So instead, we injected a retrograde Cre virus into the accumbens shell and a Cre-dependent archirhodopsin over the ventral pallidum, into the ventral pallidum. And again, by implanting optic fibers into the ventral pallidum, we were able to selectively inhibit cell bodies of the ventral archipalatal neurons. And we did this in mice that had in vivo recording arrays implanted into the accumbens. And we gave the mice sessions where they were allowed, where they had certain gave the mice sessions in this free access paradigm where there is either no laser, so just free access reward consumption, and we see this inhibition of accumbal firing. This is one example, single unit. And then on other trials, we paired this with laser in a closed loop fashion. So when the mouse engaged in a lick behavior, they got inhibition of their ventral archipalatal neurons. And it's a subtle effect that you can maybe see here in the raster plot, but it's also a bit more clear in the group data here. This is showing the normalized firing of accumbal neurons before aligned to the lick bout onset, where we see this inhibition of accumbal firing in our control mice, similar to what we've already reported and what has been reported by several others. During our laser inhibition trials, we still see this inhibition, but we've attenuated the magnitude of the inhibition by inhibiting the archipalatal neurons. And then if we look at the distribution of lick behavior, looking at the individual lick bouts, we see, again, a shift in the distribution towards more of these shorter lick bouts relative to unstimulated trials. So we are producing a shift. This archipalatal inhibition produces a shift or an attenuation of ongoing reward consumption. So what I've told you is that our archipalatal neurons inhibit the accumbal neurons to inhibit their activity in vivo and ex vivo. That endogenous activity of these neurons is correlated with reward consumption, and that driving their activity can promote reward consumption. And at this point, we weren't sure whether we'd really nailed down that this is consistent with the role of the VP in encoding value, which has been described, or could this be simply a motor effect, or a pro-appetitive effect, or a pro-licking effect? And so the last little bit of data I'll show you tried to address that really explicitly by asking, does this archipalatal activity reflect the hedonic value of reward? And we did this in a few different ways. The first was we repeated our free access experiments. This time we were filming the mice at 140 frames per second so we can measure the orofacial taste reactivity in response to the sucrose they were consuming. So this has been pioneered by Kent Berridge and Harvey Grill and has been used for decades. More recently, work by Nadine Gagola has shown that this also correlates with this hedonic facial activity, also correlates with reward valence encoding using 2-photon imaging. But essentially what we saw is that relative to the unstimulated controls, when we stimulated the ventral archipalatal neurons, this was associated with an increase in the orofacial taste reactivity score in response to sucrose, and maybe which is consistent with the idea that we're promoting the hedonic value. And maybe a bit more convincing is we did a 2-bottle choice procedure where we had 2 identical rewards. One reward was paired with nothing, so the mouse was able to just freely consume the reward. The second spout had the identical reward, but licking on the spout was coupled with closed-loop optogenetic stimulation of the ventral archipalatal neurons. And we did this in an overnight session. What you can see is a lot of variability, even in our EYFP controls, over which bottle they preferred. This is calculating the preference by volume. But in our channelrhodopsin mice, the majority of them showed this preference for the stimulated bottle. And if we break this down and look at the time course of the experiment, it becomes a bit more clear what's happening. This is a 16-hour session, and we're looking at the preference score over the course of the 16 hours. And we see that initially, in both the EYFP mice and the control mice, they will avoid the stimulation paired bottle, probably because the activation of the blue light is startling or aversive, I would imagine. However, by the end of the session, these mice are switching their preference and ultimately preferring to drink from the bottle paired with archipalatal stimulation, even though these 2 bottles contain identical rewards. And then in the final test, we head fixed these mice to remove any reward approach behavior, and we did intraoral infusions of liquids with different palatabilities. So we infused sucrose, water, or very low concentration of quinine in this head fixed task, and measured the calcium signal in the ventral archipalatal neurons using fiber photometry. And what we see is that in response to infusion onset, as we'd expect, we saw an increase in the ventral archipalatal activity in response to sucrose. We also saw an increase in response to water, but it was attenuated or to a lesser extent relative to sucrose. I should mention that these trials are all interspersed and randomly delivered, all 3 liquids within a session. And our quinine results were a little bit more nuanced, where these mice are water deprived in order to get them to participate on the task and be happy on the wheel. And initially or early in the sessions, when we're using this really low dose quinine, the mice are still drinking it. We think this is related to the hydrating effects of this low dose quinine. However, later in the session, after the mice become more sated, this quinine is still aversive, and the mice would just open their jaw and sort of let it dribble out, so they'd reject the quinine. And when we look at those trials specifically where the quinine was sufficiently aversive as to be rejected by the mouse, we see that we actually have an attenuation or a decrease in this ventral archipalatal signal. So together, this is all really consistent with the idea that these ventral archipalatal neurons encode the palatability of the reward that's being consumed. And so just quickly, by way of summary, since this is the neurobiology of addiction, we see a seminar series, we see that these ventral archipalatal neurons are driving reward consumption in a value-dependent manner. And when you think about the stages of the addiction cycle and how drugs take on this overvalued effect, or after withdrawal, where natural rewards lose their value or lose their motivational or incentivizing properties, we think that this pathway might be a really relevant pathway to target to sort of manipulate this anhedonic effect of that emerges after drug withdrawal. And conversely, when we're thinking about the glutamatergic neurons, what I told you is that they play this role in adaptively constraining reward seeking despite punishment, or learning from punishment, or titrating reward seeking. We know these neurons are very opioid sensitive, and so what we're starting to think about now is whether we could harness this plasticity by understanding how self-administration of these addictive drugs changes plasticity of these pathways. Could this be a target that we can modulate for reducing maladaptive drug-seeking behavior that emerges, and which is really considered a cardinal feature of addiction? And I will just very quickly mention that we do have open position for a postdoc, particularly looking at this archipalatal pathway and how it modulates opioid tone, and how it's modulated by self-administration of opioids. So if that sounds interesting, you can reach out. But with that, I will thank all of my lab members, really specifically Jessica Tooley, Ivan Vashez, whose work you heard about today. I didn't have time to talk about it, but Caitlin Murphy and Yuxuan Chang, a talented postdoc and graduate student in the lab, have a new preprint out looking at some of the ways we're trying to model prescription opioid abuse and understand how these circuits are affected by prescription opioid abuse. And with that, I'll thank collaborators at WashU and our funding sources. So thanks very much, and I think I will stop sharing so I can see you all.
Great talk, Megan. Okay, we have a lot of questions. The first one is from Masaya Harada, who wants to know if an individual VP neuron will receive input from both D1 and D2 in a sense, or are there subpopulations that you think get just D1 input or just D2 input?
Yeah, I think that's an interesting question. I know that others, not me, are pursuing that directly. I think with the proportions that we saw in that neuron paper, the connectivity rates, it was something like, if you're patching in the terminal fields, about 79% of the cells are getting innervated by D2s, and I think 85 were getting innervation by D1s. And so just the maths of it means that there are several neurons that have to be receiving projections from both the D1 and the D2 pathways, but how that maps on to individual, any of these functional subclasses, or whether there's other populations that are only receiving input from one pathway or the other, I don't have a better answer for that, but I know that there are people looking into it.
Okay, and then the next question is there a marker that exists for these archipalatal neurons? Is there some sort of expression? Because I think, if I remember rightly in the Malay papers, that they're non-PV neurons. I think that is like the main marker for them in the globus pallidus external. But is there anything that you've come across so far in the ventral pallidum?
Yeah, so Savio Chen had developed NPAS-CRE to label the striatal projecting neurons in the dorsal pallidus. I think it's not so clear cut. I think there are people who have looked at those and said, yes, like the NPAS neurons do project predominantly to the nucleus accumbens, but we've done some sequencing work of this population. So RNA sequencing the whole population with Mary Kay Lobo. And, she also validates that, yes, NPAS-CRE is like a really good target, but I think that there's probably heterogeneity within this population. They're sparse, but there's also neurons that project both places. And I think that trying to capture the whole population, I can't definitively say that, yes, NPAS is the way to go, but I think that NPAS is the CRE line that people have been using to target these, particularly in the dorsal basal ganglia. Right, where you have like an exclusion of the PVs. So we also, in our hands, we don't see any in our sequencing results or just our neurochemical multiplexed in situ, we don't see any evidence for parvalbumin neurons that project back to the nucleus accumbens.
Cool. As a follow-up, have you ever, this is sort of my follow-up to that, but the archipalatal neurons, they have this basket sort of projection style, and I don't remember in the paper, but do you have any like sort of anatomy of like have you ever filled an archipalatal neuron and been able to map sort of its projection field within the accumbens? I know that the connectivity seems very broad, but that's sort of you're filling all of the archipalatal neurons themselves, I guess.
Yeah. No, Kurt, I have not been able to do that, but we do have some beautiful, in the supplement, actually, we have some beautiful images. So we did our retrograde channel rhodopsin, so it's not individual cells, But it's a sparse labeling of the VP cells and projecting back to the accumbens. And Claire Lepichon at NICHD in Baltimore very graciously disco cleared them and imaged them. So those are in a supplement, and you can see this really beautiful anatomy that consistent with what Nico O'Malley described. But yeah, whether there's topography within the ventral pallidal subdivisions and whether they map on to, shell projecting or more core projecting or more, hotspot dorsal or ventral, we don't know that yet. But it's a good question.
Yeah, I'll have to go back and look at the supplement. Okay, and then I think this is from Marina, but in the thinking back to the behavioral aspect of the glutamatergic VP neurons, the session length, there was like that steep drop, like sort of after a few sessions, and suddenly it seems like they're completely shorter. What is driving that? Do you think that happens after the first? Is there like was that after the first progressive ratio session? And then there's similar like this increase in breakpoint motivation, kind of addressing both of these aspects.
Yeah, so I don't want to pull up the slide because I'll probably kick myself off this platform.
Don't worry.
So we did put the mice back in the boxes pretty quick. It was 3 days after their injection. And yeah, we're doing 3 days of FR and then a progressive ratio. So I think that it was like the 4th session of the FR. So it was after their initial, that steep drop point was after they had a progressive ratio session. So it's FR, FR5, progressive ratio, and then we went back to the FR5, and that's when we really saw the decrease in the responding. And they've had that pattern of 3 FR5s and 1 PR. That was how we did the whole testing. So it's not like they've never had a progressive ratio before, but yeah, that drop did happen after their first progressive ratio after their caspase lesion. And then the breakpoint we saw, it was more like a linear, if you actually look at the breakpoint. So right after, that post lesion, there was more of like a linear sort of increase in the breakpoint because they were tested, about a week after the lesion was their first point, and then 4 days later and 4 days later. So I think it was probably just the caspase taking time and progressively doing its apoptosis. It's our hypothesis.
Great. Okay, let me see here. We have a question. Are the archipalatal ventral pallidal neurons modulously distributed throughout the rostral-caudal axis of the ventral pallidum? Are they enriched in any given location or?
Yeah, so we did, I won't show you the slide, but we did rabies tracing to sort of look at so rabies tracing where we seeded the starter virus in the accumbens shell, either on the chat parvalbumin D1 or D2 MSNs, and then we looked at the monosynaptic inputs throughout the extent of the ventral pallidum, and we plotted as a function the starter cells or the connectivity. We plot as a function of the anterior posterior gradient in the VP, and we see it's not the most anterior subdivision, so not right adjacent to the accumbens, but really the more anterior subdivision is where we're getting more of this labeling. And I really like this question because we've had this from the Barrage group, we've had this like rostral caudal, like hot spot, cold spot definition. And with our glutamatergic cells, which is in the paper, they tend to, they're expressed throughout, but they tend to be more concentrated in the posterior and the medial VP, whereas we have this, the archipalatals tend to be, interspersed in the medial and then more anterior. So just, being a little bit too, probably too simplistic, but thinking about the functional divisions of hot spots and cold spots, that this could be one of the many substrates that goes into that functional dichotomy.
Yeah, that makes me think, I think it's like a Cronbach lesion paper where the posterior lesions make them not want to eat or drink, basically anything, everything becomes aversive.
Yeah.
Okay, sorry.
Opioid infusion into the posterior.
Yeah, is that right?
Right. I'll double check. I don't want to speak out of turn on other people's work here.
Okay, the next is sort of a question thinking about the spiral architecture between accumbens and VTA, thinking about both like the Haber architecture as well as the somewhat inspired, I think this question is by the paper from the Lamel group showing, the differences in sort of GABA and dopamine neuron innervation between the accumbens and the VTA. Do you think that such a connectivity could exist within VP to VTA projections? So maybe a lot of speculation here, but if you have any intuitions on that.
Yeah. So I'd have to completely speculate, and just from having done the recordings for the neuron paper where it was D1s and D2s into the ventral pallidum and becoming very familiar with the consistent area where you see the terminal fields, and that's not really the same area that we see with our when we do our seeding and in the same area and look for our monosynaptic labeling of the archipalatal neurons. It tends to be slightly dorsal and more lateral, those two subdivisions. So that's all the evidence I have to go on, that we don't, it's not that same area that lights up as receiving inputs from the VP as the one that is the monosynaptic input from the VP, but we've never looked at that in any careful way within the same animals trying to elucidate that tracing. But I do, and I think there's some anatomical evidence. I'm thinking back to Peter Kalivas's papers from the 90s, where he also does retrograde seeding in core versus shell and has this and circumstantial support for spiraling connectivity between these two structures, which is not inconsistent with anything we've seen, but I can't really speak with any more direct confidence on that.
So I think we have to stop there. We've had a wonderful session. Thank you very much for a great talk. Thank you for all your questions and for tuning in with us. And there'll be another talk next week, and we hope you'll continue to join the Neuroscience Addiction talks on Thursdays at this time. Thanks again.
Great. Thank you.